Good morning, everybody. Today I'll be presenting something on the role of microbiome in autoimmune. To begin with, last time I presented a detail on how the microbiota is analyzed, and there we also discussed about the various factors that actually affect the gut microbiome, and it has now been recognized as an integration center for the metabolic kind of integration and exchange for the rest of the body, and it gets affected by various factors like pollution, diet, drugs, some even psychological stresses and pathogens. The balance between the gut microbiota, the metabolism that it undertakes, and the rest of the physiology actually helps maintain the balance between health and diseases, and it is affected both by the whole group of the whole load of microbiota that is present in the gut, as well as some specific symbionts and pathogens that may come in and go out. And this has, of course, implication and response from the immune system. So when we talk about immune system, we know that there is a crosstalk between gut microbiota and liver, and bile acids play a major role in that, with circulation of almost 95% bile acids between the gut and the liver, the rest 5% being excreted. Then from the microbiota load that is present in the gut, there is a release of molecules which are identified as molecular patterns, as they are recognized by pattern recognizing receptors on the cells, and there are microbe associated molecular patterns and pathogen associated molecular patterns. Now you may ask, what is the difference between a microbe and pathogen associated, because pathogens mostly are microbes, but then not a significant difference, but the simple difference is that once it is released from a pathogen, it's called a pathogen factor molecular pattern, and generally other microbes also, they keep on shedding various proteins and structures or release vesicles which may act as molecular patterns for the receptors. In the liver, these are then captured and the immune response takes place. Till the time the immune response is active and taking care of all these patterns, and there is no excessive overload of these molecules or pathogens from the gut to the liver, everything is hunky-dory. But then when there is a loss of tolerance, which is basically the immune cells are suppressed, there is a lack of response, or there is an overt expression of these molecular patterns and molecules or pathogens which reach the liver, they kind of inflict disease, and it often ends up as an autoimmune symptom. So I need not go into the details of autoimmune disease and tell you the clinical aspects, but then for the other audience who are not very familiar with this, a quick overview that in generally, as I said, the healthy liver takes care of the molecular signals that are coming from the gut and can be pathogenic or inflammatory. They are taken care in the healthy system, but if the load increases, eventually the disease establishes, and it can go up to a serious stage also. In case of AIH, the hepatic injury is seen with interfacing with the lymphatic lymphocytic infiltrate, and this is kept under suppression, but once this is lost, it goes into progressive liver injury, fibrosis, and maybe up to decompensation. Primary biliary cholangitis is injury of cholangiocytes and interlobular ducts, and granulomatous bile duct destruction, lymphocytic infiltrate is seen in this. This is generally taken care of by, to some extent, by ursodeoxycholic acid, obeticholic acid, and fibrates that are given to patients, but who are not responding can go to progressive biliary fibrosis also. For primary sclerosing cholangitis, large and intra or extrahepatic ducts are involved. Fibro-obliterative lesions, concentric fibrotic rings, mild lymphocytic infiltrate is seen. Treatment is limited, and progressive biliary fibrosis and of the structures is seen and can become dominant cholangitis also. So coming to the interface of microbiota and autoimmune liver diseases, I'll be focusing more on autoimmune hepatitis because very limited information is available on PBC and PSC, and the main reason being that the cause effect happening in the bile ducts is not very well studied. It is more studied in the liver. So one of the recent studies has shown that there is a change in the diversity of the gut bacteria between healthy controls and autoimmune diseases as well as ulcerative colitis. But between the immune diseases, that significance was not there between the in the amount of variation that is observed in the bacteria, but between ulcerative colitis, and ulcerative colitis is also considered a kind of an autoimmune disease, although we know that there is a bacterial factor attached with it. The difference between the autoimmune and ulcerative colitis was significant. Now, the second figure here on the right, the chow richness, actually identifies the rarer species, the species that are present in very less numbers in the gut. And here also we see that there is a significant difference between the healthy controls and autoimmunes, but the whole difference between the ulcerative colitis and autoimmune was missing. So to determine what is the overall compositional difference between the gut community, they performed a simple principal coordinate analysis. But as we see from the figure here, the different disease groups and each dot showing the composition of the bacteria is pretty mixed up. It's not showing much difference. So they go on to do something called as canonical analysis of principal components, which is something very similar to a principal partial least square discriminant analysis, which we do very commonly. And they find that there is a difference in the composition of the four groups that they've taken healthy control and all that. From these, they identified that there are about 6 bacteria which are showing significant difference between these groups, and they took cohorts from two different centers, one in Hamburg, one in Munich, and they found that a good significance and area under the curve was obtained in the discovery cohort, and which was matched and replicated with the validation cohort in this, and this identifies that these 6 bacteria are able to differentiate or identify autoimmune hepatitis with just the identification of the bacteria. Anyhow, in another study, a larger group where they took 98 control samples, healthy controls, 91 autoimmune hepatitis, and the first figure A, what we see here is the whole bacterial profile between the autoimmune hepatitis and the control samples. We will come back to this figure. I just want to mention here that they look very similar, right? So how do we identify the bacteria? Still, we go into the specifics, identify different bacteria that are present in patient groups, perform some bioinformatic analysis, and what they found was that Veillonella dispar, a very common commensal in the gut, was significantly increased in the autoimmune hepatitis in comparison to the controls. But I mean, all these figures look very impressive, but we have to be very careful in observing these figures. If you look at the bottom here, there is a group of patients which are very similar to the controls, and there is no change in this particular bacteria. So the bacterial differences in relation to the patients and the stage of the patient that is coming in have to be taken up very carefully, and not just plus minus is the answer. So they go on to identify about 11 taxa which were able to differentiate very clearly between the autoimmune hepatitis and the control samples. Do not go by the minus and the plus scores at the bottom. It is basically the amount of difference that you can see from the baseline between the control samples and the autoimmune hepatitis, signifying the specificity of this, the red group of, or this pink group of bacteria to autoimmune hepatitis and the green group to the controls. So they go on to perform discovery and validation analysis in the patients. And what they do is they take different groups of these bacteria. They take one where they are comparing all the 11 bacteria. In another one, they just take the 4 bacteria. Which are the 4 bacteria? It's Villanella, Oscillospira, Clostridial species, and Lactobacillus. And then they take 7 genera and analyze by 2 different methods, Lefse and Massillin. Now, these are discriminant analysis methods developed by the Broad group of Curtis Huttenhauer, who was the first one to describe the human gut microbiome. And what they find is that the best AUC or the best results confidence was achieved with just the 4 genera that they identified here. Now, if you notice, in these 4 genera, there are species like Villanella, which was found in the earlier study also, to be increased in autoimmune hepatitis, but Oscillospira and Clostridial taxa were decreased in autoimmune hepatitis. So again, emphasizing the point that a single bacteria may not be the answer. And again, specific taxa are associated, at least this is getting clearer by this. So coming to the most elaborate and a very significant proof-sending kind of a study is from the Yale group, and they performed this study in animals which are autoimmune at birth, and then they tested that in the patients of autoimmune hepatitis also. I'll just quickly go through the results. So what they did was they took this group of animals, these are like NCW, BXSB, I'm not going into the details, are 2 strains of mice, and the F1, that is the progeny of this animal, of this pair, is autoimmune sensitive or reactive at birth itself. So they give vancomycin, ampicillin, metronidazole, and neomycin to these group and follow these animals of over 30-week period. And what they find is that vancomycin and ampicillin, they showed a clear survival benefit in comparison to the other drugs. Now, both these drugs are specific for gram-positive bacteria, broad spectrum, but gram-positive, so which is an indication that some gram-positive bacteria is involved in this suppression of the autoimmune signal. They followed this up with the identification of the MAMs and the PAMs that I discussed earlier. So double-stranded DNA is a DAM basically. So what they see is that with vancomycin, the anti-double-stranded DNA antibody was reduced in comparison to control or neomycin or metronidazole. Same thing for the anti-RNA antibody. It was decreased in comparison to the control animals and the other drugs. They also did check about the leakiness of the gut by saying that, okay, some pattern is being generated from the gut has to have a breach in the barrier, and the products, intraluminal product, should leak out through the portal vein and reach the liver. And what they did was they used a FITC labeled dextran solution gavage into the animals, and what they find is that again in the control group of animals, another strain of animal 57 black 6, which showed a strong gut barrier and a poor translocation of this compound, this labeled compound. In the autoimmune, it was severely increased, but in the vancomycin and ampicillin group, it was reduced significantly. Again, confirming that there is a breach in the gut. And that is why these DNA and RNA are probably reaching the liver. If they are reaching the liver, they should be present during the pathway of the transit also. So the mesenteric veins also showed that the bacterial count was low in vancomycin. Of course, it was suppressed, increased in controls, and absent in the control animals, confirming that yes, it is pathogen related, what these signals are that we are seeing. Then mesenteric lymph nodes, known as the first firewall after the gut, they also showed a decrease in the vancomycin ampicillin group as far as the bacterial counts are concerned when they were cultured. Now, in the liver tissue, the same results were observed. Control showed that there is bacterial load as shown by the CFUs from the liver tissue, not present or minimally present in vancomycin or ampicillin group, clearly showing that there is a suppression of some kind of bacteria. So what they do is they take the hepatic isolates, culture them, perform a 16S RNA sequencing to identify which bacteria these may be. And what they find is that there is a species called as Enterococcus gallinarum, which is showing an increase. So they say, okay, if this is present, then we should see this species, this bacteria in the histology also. And what they do is they take two groups of animals. One is the germ-free, one where this gallinarum species is present, and they stain it with antibody against the gallinarum. And what they see is that it is absent in germ-free animals and is showing in the liver tissues, shining as these specks that we see here, confirming that the bacteria is present. Very interesting. So the whole bacteria is moving from the gut, from the breach of the gut into the mesenterics, into the liver, and sticking around. Then again, of course, gut barrier integrity is lost in this. That is why it is reaching there. So they wanted to confirm if this is this bacteria specific, or any bacteria can actually go and induce this, can be placed in the liver. So they use another, this is Enterococcus gallinarum. They use Enterococcus fecalis, another commensal bacteria present very commonly, and Bacteroides theta iota micron, another bacteria that is present. All these were present in the same animals at the same time under normal conditions. But when they perform the RNA-seq analysis of the liver, of the ileal tissue, what they find is that there is a significant change from the rest of the group in the Gallinarum species enhanced animals. So they go on to confirm if there is a breach in the liver. They check for zonulin, occludin, mucin, claudin, PLVAP, and axin to see if the integrity is maintained or not. So what they find is that in the Gallinarum group, this is significantly increased in comparison to the other bacteria or the germ-free animal. GF is germ-free animal. The occludin, although, was reduced, which means there is a specific type of injury happening in the gut also, which is not a pan tight junction protein suppression, but selective tight junctions are getting suppressed. Mucin did not show a significant difference between any of these bacteria. What they have not checked is a species called as Akkermansia mucinifila, which is, as the name suggests, feeds on mucin and kinds of maintains the homeostasis in the mucin levels inside the gut. So had they shown that, it would have been much more clearer on what is happening with the mucin levels. Claudin 3, again in Gallinarum it is increased, and the other two proteins of the cellular integrity do not show a difference between the groups. So what they did next was very interesting. They take the hepatocytes and they co-culture these hepatocytes with the bacteria that they just tested before. And what they find is that they take the whole bacteria as well as they take the RNA from these, the total RNA from these bacteria, and put it in the culture. So the hepatocyte is also growing, and you have all these pathogens and pathogen-related molecules. Now, for some reason, they have to tilt the graph 90 degrees to the right, so we have to look it this way, increases moving towards the right. So beta 2 glycoprotein 1 is increased in the cultures where the cells were present. It is not increased in cultures where the RNA was given. Okay, I'll come to what this is doing. So basically, hepatic glycoprotein GP, this beta 2 GP1 is the one which actually once released in the circulation leads to antibody generation, and which can eventually lead to thrombi formation. IFN alpha, IFN2, we see that there is a significant increase in the cells as well as the RNA that was present with the hepatocytes, which means that hepatocytes are responding to both the cells and and this is only hepatocytes, there are no immune cells here. So hepatocytes are releasing interferons in presence of the bacterial cells as well as the bacterial components. And AHR, which is a xenobiotic receptor, also shows an increase with the gallinarum cells and RNA. So confirming that hepatocytes are responding to the bacteria that are reaching the liver, releasing inflammatory molecules to which the immune cells respond. And I'm sure you must have formed that image that once the immune cells are reaching the hepatocytes, they're automatically attacking the hepatocytes and destroying them, leading to autoimmune disease. But okay, this is in animals. Another protein that they found was endogenous retroviral glycoprotein GP70. Now this is a retroviral plasmid protein, a fragment of that which got inserted in the human genome sometime in history and is persisting, but has integrated so well that it is involved in intracellular vesicular transport now, which is through the Golgi body complex. So what they find is that there is an increase of this protein GP70 in the animals as they age, which means that of course they are autoimmune at birth, but as the disease progresses, the concentration of this molecule starts to increase in the circulation. And what they also found was that the increase of this molecule is directly related and significant with the TNF-alpha expression in the livers. So of course this is present in the genome being expressed in the different tissues, also livers. Okay, very good. So if this is antigenic, then there should be antibodies for it. And this was proven that when antibiotics are given, the antibody concentration of this particular protein reduces. So what is concluded from here? So this is the genome. So the bacteria is going to the liver. What they have not shown is how it is actually triggering the expression of this particular gene or this protein. But this protein gets activated, is released, TNF-alpha levels go up, but when antibiotic is given, the bacteria is not going to the liver and the amount of protein is also reduced. So there is a correlation here between the bacteria and the antigenic molecules. Now, what they go on to check in patients is that they have used what they call as normal healthy donors. I'm not sure how they differentiate healthy donors from normal healthy donors, but normal healthy donors, systemic lupus and autoimmune hepatitis, and what they find is that this GP70 or endogenous retroviral protein has a significant increase in the antibodies in lupus and autoimmune hepatitis, both being autoimmune disorders. Again, anti-human RNA antibodies were also found to be increased in these groups in comparison to the controls, which sets a direct correlation that this protein is involved in autoimmune induction. Anti-human RNA IgG was significantly correlated with the anti-EG RNA that was being expressed. Of course, R square was 0.51, but in autoimmune hepatitis patients, R square was 0.8 and was much significant when the anti-human RNA, anti-RNA IgG was compared with the amount of RNA, which goes on to show that a persistent increase or persistent release of this protein is eventually going to bring the immune cells to the liver and inflict injury there. So after this, there have been other studies where fecal cytokines have been used to identify if instead of going to the bacteria, doing next generation sequencing, which takes time, if there is a cytokine level test that we can very easily do in the stool samples and identify if there is a difference between the controls and the disease groups. So the white boxes here are healthy controls. The light purple are the sclerosing cholangitis, and the dark purple are the autoimmune. As you can see that as the disease becomes is more severe, maybe in case of autoimmune here, we see that in the fecal as well as plasma samples, there is an increase in IFN gamma, IL-17, IL-1 beta, IL-6, TNF, IL-8, but in the fecal samples, the IL-10 was not significantly different, which means that the immunosuppression signals are not working very well in the gut, but activation signals are working much better in these setups. This slide I've taken from Dr. Sukriti, and they were performing some experiments on autoimmune hepatitis stool samples, and they also found that IL-17 was increased, though not very significantly, but IL-10 was definitely suppressed, which was meeting the results, matching the results that were found in the previous study. IFN gamma, on the other hand, was significantly increased. So again, confirming that the inflammatory signals in autoimmune are running in parallel, or maybe start earlier in the gut itself, and I don't know if it is significant clinically or not. If the patients are followed for their gut profile, gut inflammatory analysis is done for all the patients, maybe the future of liver disease can be correlated to some extent. Of course, detailed studies are required on this. So to summarize, the gut, in the gut there is a barrier breach which leads to release of the specific pathogens. Here we have seen that Enterococcus gallinarum is there, but these are American studies, and as I've shown earlier, there is a difference in the gut microbiota of the Indian population and the American population. So we have to identify our own species. It may or may not turn out to be the same. This leads to release of specific proteins which not only increase the inflammatory molecules, but then also lead to formation of antibodies which inflict injury on the liver itself. There have been many studies. I have not included most of them, and these two slides are the summary of what is being found in autoimmune hepatitis, PBC and PSC. But interestingly, most of the studies have performed a reverse kind of analysis. They've taken the patient and seen what are the changes in the gut, which of course we understand that and has been proven many times that with liver injury there are changes in the gut microbiota composition. And there are so many bacteria which are occurring repeatedly. Villanella is there, Streptococcus is there, Pervotella is there, Klebsiella is there. Interestingly, Klebsiella is increased in autoimmune hepatitis, but in PBC, Klebsiella not only is increased, but in the individuals who were increased had poor remission and did not show good progress with the treatment. In PSC, of course, there are many studies and various samples, right from bile to colonic mucosa to feces to oral sample saliva have been taken. Two studies have included fungi, role of fungi, or try to understand how the fungi change in the gut. Again, this is not the cause and effect, but with the disease, how does the gut microbiota or the gut fungi change? And what they find is that Humicola and Candida Humicola is a fungi which actually consumes humic acid that is being produced transiently in the gut, and Candida, they show an increase, but overall fungal diversity did not change. Now, just as a brief touch on, the amount of fungi that is present is just about 1/10 or 1/100th of the total bacteria, that is the number of cells that is present, but occupies almost 30 times more space because of the large size in the gut. So the space occupied by fungi is more than it is occupied than by the bacteria. Anyhow, so the diversity did not change. So I try to summarize these three tables, and what we find in autoimmune hepatitis is increase in Villanella, Streptococcus, Lactobacillus, Akkermansia faecalibacterium, as well as increase in the oral bacterial diversity. That means overall the number of species in the buccal cavity, it increases. Now, all these bacteria that we see here are under healthy conditions present in the gut. But concurrently, there is a decrease in the gut diversity. Oral diversity is increasing. Food is going through the mouth into the guts. We are drinking water, so bacteria is translocating, but it is not leading to an increase in the gut diversity. The gut diversity is suppressed. Pseudobutyri vibrio, Lachnospira, Prevotella, Parabacteroides are decreased, but oral streptococcus, which was increased here, is decreased in the gut. We expect it to move to the gut, but it is decreased. In PBC, many species that are present in AIHS are also found. Besides, Haemophilus, Klebsiella are also increased, and Klebsiella, as I just mentioned, is associated with poor remission. Other bacteria are decreased in PBC, which have been shown to increase with ursodeoxycholic acid administration. In PSC, Faecalibacterium, beneficial bacteria, Roseburia, Alistair Borachia, all these are beneficial bacteria are increased. But in contrast to autoimmune hepatitis, in PSC, oral diversity is reduced. So maybe if we can develop a quick test on the total microbial load present in the buccal cavity and establish a cutoff in comparison to healthy, we can very rapidly test if the patient is a PSC type or AIH type right when the patient comes for the first time. Now, so this is all I had to say about the AIH, but then I'll touch upon something which is very important for any disease. I'll take an example of this figure that I showed earlier here also, that they have taken 91 AIH and 98 controls, and this figure looks, or these are the 10 phyla that they are comparing here, and this figure looks very much similar. But this is when they have performed the study, taken all the patients and analyzed the data. Now, what if the patient is coming in and you have a certain bacterial profile identified, and some data is missing? What we see is that they are exactly identical. It is very difficult to even identify this person is healthy and that person is here only because he's saying I have a pain here or pain there. The other alternative is that we have taken less number of samples. Now in this, if we look at the left and the right sections, we see that there's a very, very low diversity of Bacteroides in this and a very, very high diversity in the AIH group alone. Same thing we see in the controls also. How do we differentiate whether this control is eventually going to become an AIH or not? We cannot. So sample size in the studies is very important. Elaborate analysis is definitely much required in any kind of microbial analysis. Added on to this is when this group, the Yale group, compared different diseases, cirrhosis with autoimmune hepatitis, and what we find is that this bacterial profile appears very similar in comparison to controls. And this red group, which is nothing but Enterococcus, seems to be increased. So just an increase in Enterococcus does not indicate autoimmune hepatitis or does not indicate cirrhosis. We have to go into little more details, identify more specific parameters. So currently we are in a very early stage of microbiota analysis and its relation to liver diseases. But yes, work is going on, though at a slow rate. So to end the presentation, what I understood from all this study is that there is a significant association of specific gut bacteria with autoimmune liver diseases. Bacterial cells and metabolites migrate to liver and induce inflammatory response. Additional damage to the host tissue in response to hepatic bacteria occurs as the immune response increases, and the gut microbial profile varies between the disease groups. That should be analyzed and interpreted very carefully. Thank you.